In this video, I'm going to give you a quick introduction on how to use GenePool. GenePool is a cloud-based software environment developed by StationX that enables users to securely manage, analyze, and share human-derived genomics data. In order to log into GenePool, you can go to www.stationxinc.com and click Sign In at the top, or go directly to https://stationx.mygenepool.com. I'm now going to log into my GenePool project account. Once you're logged into GenePool, you'll be presented with a dashboard view that gives you a sense of your recent activity in GenePool, and quick access to the help and support portal where you can ask questions and find documentation. For instance, if I wanted to look for documentation on how to import data, I might search for data import. You can also launch into the import data workflow from the dashboard. You can also take a look at samples that are already imported into your account or available to your account through the Genomics reference library that is available to all users. Let's click into the data library and take a look. There are two main areas once you click into the data library that you'll see. The first is a reference area which contains projects that are available to all GenePool users, whereas My Projects contains projects that are only available to the user that is logged in. In this case, this is data that is only available to me in my account. When I navigate the folder tree structure, I can see a number of projects that are available to me in the reference library. And if I click on any one project, say the breast cancer project under the TCGA expression folder, I see in the right frame 1100 RNA-seq samples populate along with all their metadata that has been attached to those RNA-seq samples. Likewise, I can click on, say, a sequence variation project and see, like the bladder cancer project, I can see 194 samples that have been imported into GenePool. This actually represents 97 tumor normal pairs that were generated as exomes and called mutations through a somatic mutation caller. If I want to work with any of these projects, all I have to do is click on one of the projects and then click Analysis tab. The alternative route to get there is actually to go through the dashboard. Click on Start a New Study, select a project, click on the project of interest, say the prostate cancer RNA-seq project in the reference library, and then take a look at the various workflows that are available to you. Under the analysis drop-down, you can see that there are a number of workflows that are available. Since this is an RNA-seq project, I have a number of expression workflows available to me. Now I'm going to try the expression comparison workflow. The other parameters are defaulted to follow best practices, but you can change them if you so desire. I'm going to leave the defaults on and just run the carcinomas as group 1 versus the normals in group 2. Effectively setting up 20 samples of carcinomas versus 10 samples of normals and running a differential expression analysis. By clicking Run Analysis, GenePool is going back to the database, retrieving the RNA-seq counts, computing the raw gene counts, and then doing normalization across the samples, and then computing differential expression of 20 versus 10 as designed by the way we did sample selection. You can see here GenePool returns a view that contains interactive visualizations along with the table view containing various statistics as well as a number of annotations. You can navigate and filter by taking advantage of column filters in the table, as well as adding and subtracting columns through this drop-down menu. You can take advantage of these interactive visualizations to filter for groups of genes, For example, I can use the volcano plot here to select for the most differentially expressed genes by p-value and by fold difference. Keep those. And you can see I've reduced this down to the top 875 genes by p-value and fold difference. You can click into various views, such as the enrichment analysis view, to take a look at which gene sets are most enriched. You can click into the heat map, for example, and take a look at the top genes and how they express across 30 samples. You can navigate the tree, the dendrogram, as well as filter. You can also go into the co-expression network view and filter based on correlation coefficients computed as gene by gene Pearson correlation coefficients. And lastly, you can actually sort. You can do things like sort the table and take a look at the gene expression for any one of the genes across samples. For any of the tables you see in GenePool, you can actually export the results as Excel files or tab delimited files, or you can actually save the results within GenePool, or save the query that got you there, such as the analysis setup that you saw when we set up the 20 versus 10 samples, so that you can come back to GenePool and rerun that query at any time. Why don't we take a quick look at running a sequence variation analysis. As we just run an expression-based analysis, let's take a look at running a mutation analysis with VCFs that have been imported into GenePool. Let's take a look at a small project. We'll look at the Ceph trio with 3 samples. These are exomes that have been imported in from VCFs for these 3 samples. I'm just going to run a simple variation profile while there are a number of other analyses that are available, such as variation comparison. If you wanted to compare, say, subpopulation of tumor VCFs versus another population of VCFs. You can also do trio analyses if you identify the parents and the child and run a trio analysis. But I'm just going to run a simple variation profile. Select all 3 of these samples, drag them into the target, call it a Ceph samples. You have the option to change some of the minimum thresholds for quality, coverage, and variant frequency. I'll leave the defaults on. Run the analysis, and what this is doing is actually going back to the database, retrieving the mutation calls, and dynamically annotating those mutations with various tags. And we can take advantage of those annotations now and actually filter for interesting mutations in one or more of these 3 samples. For example, if I wanted to look for non-synonymous mutations or those kinds of mutations that are likely to have a biochemical impact, say frame shift mutations, splice site effectors, I can open up this column which shows you the effect as well as the impact as computed by SNPF. And you can see that things like moderate encompass non-synonymous coding events and some other events. You can see things like high impact mutations tend to be things like splice site acceptors, as well as other categories which include frame shift insertions and deletions. So we can take advantage of widgets to do that. So I'm going to use this particular widget here and select for the high and moderate impacts. You can see that there's going to be about 35% plus 1%, 36% of the mutations here actually meet that criteria. Keep those mutations. And then I can actually select for rare mutations in this data set by, say, adding the 1,000 genomes allele frequency widget. I can select for those mutations that are unannotated in the 1,000 Genomes project, so likely either very rare or just haven't been seen in the 1,000 Genomes project allele frequencies, and also those that have been seen and have been seen at 5% or less. So by using the widget, I'm actually selecting for the rare mutations across these 3 samples, these 3 data sets. And then I might use something like ClinVar as an example, as another database, and look for those mutations that are known to be pathogenic. So what I might do is remove all these mutations that do not have any annotations in ClinVar, and then take a look at those that do have annotations in ClinVar, say the pathogenic ones, as opposed to the non-pathogenic or those that are just histocompatibility associated mutations or untested. So it looks like 16 of these mutations have a pathogenic potential. So I'm going to keep those. You can see 16 mutations meet that criteria. Why don't we take a look at all those mutations? So you can see here that within the table there's a number of various statistics and annotations available, such as the fraction of samples that's been seen in, the allele frequencies, genotype calls, coverage, and various annotations. So as a reminder, you can add or remove any of these columns, and of course save the result within GenePool, save the query that got you here, as well as export out to tables. As an example, you might also want to take a look at one of these mutations and verify its presence in the sequencing reads. So if I select, say, this mutation in SYNE2 that is associated with this particular disease, according to the clinical genomics database, you can also open up the annotation for ClinVar. You can see here that that particular disease is associated with that mutation in ClinVar, as well as the clinical genomics database. I might go and take a look in the browser. And in GenePool, the browser actually pulls reads from a cloud storage solution. In this case, the samples are being pulled from an Amazon S3 bucket where the BAM files are stored for these samples. And you can see here that there's a coverage plot available for over this mutation. And you can see that the coverage is around 70x across these 3 samples. For these 3 samples. If I click on this variant track here, I can get a sense that 2 out of the 3 samples have the mutation called. And I can see that these are the 2 that have the call, while this is the one that does not have the call. So why don't we take a look at the sequencing reads for one of the samples that has the mutation call, whereas this sample here does not. And these reads are, as I mentioned, are being pulled from the Amazon S3 bucket for these samples on the fly. So that's a general overview of GenePool. There's a number of other things that are available, and if you're interested in learning more, please check out some of the other videos that go into more detail about each of the individual analysis workflows, as well as how to import data and a number of other things that you can do in GenePool.